Dear students, we move on. Tunaendelea kutafuta banda wanafunzi wangu. So, we talked already about lipids, and we started with we already mentioned few functions of some important lipids, and to be specific, we talk about the phospholipids. But phospholipid is not the only type of lipids that we are supposed to know. We talk about fats as well, and functions of the fat. We say that we have a concept of saturated and unsaturated lipids. We add more to our lesson, and we talk about steroids. So what are steroids? Steroids, they're basically also composed of 4 carbon rings. They're types of lipids that are also composed of 4 carbon rings. And most of the animal cell membranes, they do contain steroids in form of cholesterol. So here we have one more function of lipids, and to be specific, cholesterol, they are found in cell membranes. They form together with phospholipids, integral parts of what? Of the cell membranes. I'm getting more curious about the structure of the cell membrane. I want to see how it looks like. So, tusubiri, tutakuja kuona vizuri katika wakati tutakaposoma kuhusu cells, na tutaona phospholipids na cholesterol wanatokea vipi kwenye cell membranes. So, what are other types of lipids? We have prostaglandins. Prostaglandins, they are also types of lipids. They're basically modified fatty acids. And what function do they have? They do act as local chemical messengers in our bodies. What is that? They are used in cell communication. They are used in cell communication, and to be specific, they are actually playing a very important role in inflammatory reactions. Excuse me, it's not K, it's C. I have problem with English and German here. Yep, inflammatory reaction. Okay, but very important, we also have some hormones which are made of steroids. So when we talk about steroids, I also wanted to say that we have very important hormones which you already have heard. I'm sure you already have heard of them, estrogen and testosterone. They function as hormone in the human body. So here we have very important roles of lipids. We have mentioned about their function in the cell membranes. We have mentioned their role as hormone, and as well in the inflammatory reactions, or as local messengers in the cell communication. But as well, we have other forms of lipids, and that is now to be specifically fats. Of course, we mentioned about fats, but what are the roles of fats? My dear students, please keep it in your mind. We know that the source of energy, the basic source of energy, or the common source of energy in our body is glucose, and that comes from carbohydrates. Yeah? Cool. But you have to know that most of the fats, they contain very long carbon chains. Basically, the ratio of the energy storing carbon hydrogen bonds that are present is twice more than that of the carbohydrates. The carbon hydrogen bonds that are present in the fats, they make them efficient storage form of energy. And in this manner, we say that when you break out one molecule of lipid or fat, you expect to get more energy or more calories as you would get when you have broken a carbohydrate. So we also mentioned about the concepts of saturated and unsaturated fatty acids already. You know that most of the fats that are produced by animals, they are saturated. Why? We mentioned already about that, because they have all the carbons saturated with hydrogen, and therefore they cannot easily bend, the molecules there cannot easily bend on one another, so they are not oils, but they are solids. But we also have plants which produces fats, and plant fats, they are basically unsaturated, so they are most of the times oils. Nadhani mnajua kuhusu peanut butter, inapatikana kwenye maduka, inakuwa na karanga ndani yake, na mnajua makaranga yana mafuta. So wakati mwingine yale mafuta yanaweza kujikusanya katika kile chakula ambacho kinatakiwa kile kibaki kama solid, ili kiweze kupaka kwenye mkate ama kitumike kwa namna nyingine. So wakati mwingine kuzuia yale mafuta ya mimea yanayokuwa katika chakula yasiweze kujirundika kama mafuta. So what the scientists, they have discovered is that there is a concept of artificially hydrogenating those unsaturated fats to make them solids, or to solidify them. Yeah, tuna add more hydrogen, so zile fats zinakuwa then hazijikusani kama mafuta, na hivyo inaonekana vizuri, lakini that seems to eliminate the health advantage they have over saturated fats. What is more important is that unsaturated fats, they are healthier as saturated fats. That is something that I don't want you to forget, my dear students. Yeah? So tunapokuwa tunataka kupreserve chakula, kukifanya yale mafuta yasilimbikizane katika chakula, kama maji tukayasema tunayasolidify kwa kuadd hydrogen, of course what we are doing then is changing unsaturated fat into saturated fats, and that makes it both equally rich in carbon hydrogen bonds, and therefore they are no better than any unsaturated. Yeah, so we have something else here to talk about, my dear students. Fats, although they are good storage, energy storage depots, and they have very good things, we have talked about very good things of advantages concerning the fats, but not everything is always good. You can see here a very sad face. Yeah, accumulation of lipids in our body has also some harmful effects, and to be specifically, when we have lipids accumulating in the layers of blood vessels, they have endothelial cells, right, dear students, where here blood is supposed to flow smoothly. All right, so when we have these very beautiful looking molecules, but dangerous, they're looking good, but they're dangerous. These are cholesterol types. They're type of lipids. When they accumulate in the blood vessels, my dear students, they can then reduce this caliber here. You see, the caliber of the blood vessel is reduced, and this can really disturb flow of blood. So instead of having a smooth flow of blood here, when then the too much of the cholesterol has deposited itself in a sense that this blood vessel dimension is reduced like this, then it is actually not good. So there's a concept of atherosclerosis. We call it atherosclerosis when we have so much cholesterol accumulating within the layers of blood vessels. Why is it not good? Because it can lead to complete obstruction of the blood flow, and it could cause heart attack. Oh my god, this is a very sad story. Umekula mafuta, end of the day umekuwa na umri mkubwa, yanajimbikiza kwenye nini? Kwenye mishipa ya damu, yanapunguza ule uwazi wa mishipa ya damu, then they can block completely, especially ile mishipa ya damu ambayo inakuwa ina supply damu kwenye kuta za moyo, kwa sababu moyo wenyewe licha ya kwamba una pump blood kwenda kwenye system za mwili, unahitaji pia damu ili uweze kuishi ule moyo. Kwa hiyo ile mishipa ya damu ambayo iko nje, ya ile mishipa ya damu iliyokuwa nje ina feed muscles za moyo, inapo block, then we have a problem. That means the muscles of the heart cannot get enough oxygen, and that would lead to damage of these muscles. So basically that's what is happening when we have a heart attack. So, but my dear students, something else can also happen. That's a bad story, a very sad story happening when you have this. When you have, we already said that we could have cholesterol accumulating in the blood vessels, right? To decrease the caliber of the blood vessel. Caliber means ule unene. So kama hizi cholesterol zikidetach, if they do detach, yeah, if they do detach from their attached positions, then hii sehemu ambayo imedetach, yeah, inaweza kuactivate certain reactions katika mfumo wa damu, kusababisha hapa damu kuclot, damu kuganda, damu inaweza kuganda hapa. Kwa hiyo pia hiyo inaweza ikasababisha blockage ya nini? Ya flow of blood. These are two different concepts. The accumulation of cholesterol here katika mshipa wa damu kusababisha heart attack ni different concept na cholesterol plaque tunaita, ile mkusanyiko wa cholesterol, alafu yale mafuta yakajipakua, yakajiachia ile sehemu ambayo mafuta yamejiachia, inakuwa ni kama vile inaweza kuchokoza damu kuganda. Kwa hiyo reaction itaanza pale, then we can have clotting of blood. And imagine something like this happening in a blood vessel which is supplying part of your brain, my dear student. You could have a very big problem, and that is a reason why we have stroke. We have strokes. Actually, my beloved students, strokes and heart attack, they are basically similar concepts, in a sense that in both ways deficiency ya supply ya oxygen inasababisha zile cells kufa. So in the case of heart, then blood muscles of the heart, they lack blood, so they start to cry out of this lack of oxygen, and then a patient is experiencing pain, has chest pain, something like that. But when blockage of blood vessels occur in a brain, then basically those cells of the brain, which are very sensitive, they would lack oxygen as well. So when they lack oxygen, they will also start to complain, and that's what we call stroke. Mgonjwa wa stroke anapodevelop hiyo event, haraka sana utaanza kuona vitu vya haraka pale vinatokea, kama misuli yake inapoteza nguvu, anazungumza kwa matata na vitu kama hivyo. Kwa hiyo hiyo ni hasara pia ya nini? Ya accumulation of cholesterol, or disadvantage specifically happening due to atherosclerosis. So atherosclerosis ni kule kukusanyika kwa zile cholesterol katika mishipa ya damu, my dear students. So now we are done with that. Let's finish our lipid section by talking a little bit about food tests. I prefer to explain it to you here in theory lesson, because nowadays you're being tested in your theory exams. You have also questions from the paper 3, but even if that not the case were, you have to know about food taste, because you will eventually have to do this practical exam as paper 3, right? So how do we test fats? We have basically 2 types of tests, and one of them is emulsion test or ethanol test for fats. What are we gonna do there? We have our food sample. We have our food sample to be tested, and to this food sample to be tested, what we are going to do is we're gonna add ethanol. Alright, students? We add ethanol, and then you can say, let's say 2 cubic centimeters you can add there, and then you shake it. After you shake it, my dear students, you let it settle in a test tube rack. You let it settle in a test tube rack, this mixture. You allow it to settle for 2 minutes, and what's gonna happen is that a milky white emulsion will be formed. Oh, super. What is that? What is happening here? Unaona ile cloudy solution inaanza kutokea pale. That is called emulsion. So basically, my dear students, when when something like that happens, we can say lipids are present. Lipids are present. That means that solution does contain lipids. So haraka, what happens there is that we know that lipids, they are insoluble. We did talk about it at the very beginning of our session. So we are taking advantage of this property of lipids being insoluble. So we add ethanol to it. When we add ethanol to it, they will dissolve. So lipids will dissolve. Yeah. Students, sorry, I did forget something very important. Yeah. Okay, so I will repeat quickly. Yeah, just to catch it up. We did have our food sample there, right? And then we added ethanol to it. Then we added water. We had to add water. So when we added ethanol and shake it, the solution was really clear, and then we let it and then we add water and let it settle in test tube rack. That is when you observe formation of a cloudy layer or emulsion. Yeah. So that is that is the first point about the emulsion test for lipids. Then we have the second type of test which we use to see if we have lipids in a solution or not. And that one, my dear students, is called Sudan 3 test. What we do in Sudan 3 test is at the beginning somehow similar to what we did by ethanol test. So we have food to be tested with water inside it. Yeah, equal amounts of food and water, they are added to a test tube, and then you add very few drops of Sudan 3 solution to your test tube containing food sample to be tested. So Sudan 3 is to be added to a solution to be tested, and then shake it. After you shake it, my dear students, if you see some droplets of lipids above, forming a layer above, just above, on top of the solution, forming a layer there, we can say lipids, they are present. It means Sudan 3 is going to stain the lipid molecules which are present in your solution. Oh, my dear students, I love biology. So kwa leo tuishi hapo. We will see each other next time, and we're gonna talk. Our next step is to discuss about carbohydrates. So dear students, don't worry about the notes. You will have notes Utapata notes nzuri kutoka kwetu na utaweza kujisomea kutoka kwenye hizo notes, na basically itakuwa ni yale yale ambayo nimeyazungumza kwenye lecture zangu. Wanafunzi wangu, napenda kuambia kitu kimoja. Sisi tuna wasaidia kuweza kuwafanyia haya masomo yawe mepesi, but sisi tunawapa percentage ya kile ambacho mnahitajia. Juhudi zenu ni muhimu sana, ni muhimu sana katika kufanikiwa kwenu. Masomo yenu ya A level especially wale ambao wanachukua PCM na PCB, to my experience, wanahitaji kuonesha juhudi zaidi, especially wanaosoma PCB, because biology needs a lot of your time. You have to study again and again, and since you wanna maybe become doctors or you want to work in pharmaceutical industries, or you want to become dentists, or you want to work in lab, you have to have a very good concept. You have to grasp this biological concept very well. Kwa hiyo, keep learning, but work really hard. You have to study by yourself as well. We talk more about how you can manage to study and different techniques of learning. So dear students, see you soon. Take care of yourself.